Okay, so I actually, did we talk about 21? Okay. Well, we're a little bit. We are recording. Okay, so there are ways that cells interact with one another. One of those is a desmosome. And we talked about the idea of rivets. We have buttons and things that we think of as rivets, where you take two pieces of metal and kind of squish them together from either side. In this case, you're riveting things in place so they don't move. And these desmosomes act as anchoring junctions that actually prevent cells from being pulled apart as a result of mechanical stress. Okay, it's kind of like a button-like thickening of adjacent plasma membranes. Now, why would you need something like this? What kind of mechanical stress might these kinds of cells be under? If you're unsure, just take one of your cells off and see what happens. Yeah, so I mean, are you able to just like grab a cell? Why not? Is there a resistance to you pulling away from your skin? I mean, you move all the time. Why don't you? That sounds odd, but if you've ever seen like movies where people just like disintegrates into bubbles of, why don't you do that? No, they're anchored together. Okay. Yeah, exactly. Okay, awesome. Did you turn in curiosity?
No, I seen that.
I reopened it
Yeah, I seen it.
for tonight. Okay. Yeah, so just make sure. Okay. There are also gap junctions. I think I briefly mentioned this last time too. This one's for allowing communication between cells. The little hollow cylinders of proteins that when two membranes come together, it kind of allows a little space for them to travel, things to travel back and forth. So molecules actually directly travel from one cell to the next through these channels. And I don't know if y'all have seen, never mind, I'll just ignore that for the moment, but probably you may have even seen commercials that show this kind of gap junction when they talk about like medications that help with what's the word? Oh, goodness, it just left me. So when you talk about serotonin and acetylcholine and things like that that are helpful for kind of mental acuity and mental health and just basically getting your brain to think straight. Those are, you may see these little models sometimes on the commercials that show gap junctions. That's not a good example. I'm sorry, guys. But I'm just saying, you may have actually seen a representation of this. So you can see the tight junction at the top. Okay, it's impermeable. I've just covered it up. Whoops, go back. There is the desmosomes, this anchoring junction that acts like little rivets. And then you've got gap junctions that actually allow things to go between one and another. Okay. So your cells are not individualized. I think that's the thing to maybe take from this. As a multicellular organism, your cells work together. Okay. Your cells function not individually, but as a group. And so they anchor themselves together and they communicate. They have to have ways to have communication between those two cells. And so this is direct communication. There are other forms as well, but this is cell-to-cell communication between these gap junctions. So what is inside the cell is an area of that we call cytoplasm. It's a cellular material. It's outside of the nucleus because we called it nucleoplasm inside the nucleus. But it's inside of the plasma membrane. Most of your cellular activities take place within the cytoplasm. This includes the cytosol itself, inclusions, organelles. What I want you to know from this is that the cytoplasm is going to be basically whatever else is inside the cell that's not in the nucleus. Okay. The cytosol is the fluid that suspends all these other elements. Sometimes I feel like people use the term cytoplasm and cytosol interchangeably, but that's not the case. The cytoplasm is more inclusive than just cytosol. The inclusions are chemical substance, just like either stored nutrients or cell products that have been made and are floating around. And then you have organelles. Okay, so this is your machinery, the metabolic machinery of the cell that does all these different functions. So anything, well, not all, there are a few that's not necessarily membrane bound, but most things are membrane bound. That means they have their own membrane that either separates their insides from their outsides, or the membrane acts as a separation from different areas within the cytoplasm. Can y'all name a membrane-bound organelle that you may know about inside the cell beyond? Sometimes nucleus gets involved there, but I'm really not focusing on the nucleus. It's really separate from that. Anything else? Just name something inside the cell, and I'll tell you if it's a organelle or not. Seems like this. Yeah. And I'll tell you if it's an organelle. You know, if anything you can think of beyond nucleus is supposed to be inside a cell. Mitochondria. Okay. Mitochondria, by the way, that's the plural. We have multiple mitochondria. A single is a mitochondrion. And a mitochondria is all about, it actually has two membranes, and it's used for making ATP. So can you think of something else now that you've kind of broken the ice? Got all the famous ones. No, nucleotide is going to be a monomer of the nucleic acid, which is your DNA. Now, could those be floating around in there? Yes, but those are probably would be considered inclusions as they were made. If you've got free floating ATP, which would be a nucleotide, probably would be considered an inclusion. Now, would they be present? Absolutely. Just not a membrane bound thing. So it's not the organelle. Have you heard of endoplasmic reticulum? Okay, it's sometimes called the ER. You'll sometimes also hear it called smooth ER and rough ER. Does anybody know why one is smooth and one is rough? What's the rough? The ribosomes. Okay, ribosomes are considered, can be considered an organelle, but they're one that doesn't have a membrane. Okay, so they're not the membrane-bound versions. But some people do consider it to be an organelle. How about the Golgi? Golgi body or Golgi apparatus? Okay. There's also something called a peroxisome. We'll look at some examples now. But the ones that most likely get talked about are going to be mitochondria, Golgi, ER, endoplasmic reticulum. Those are your kind of your big ones. So here's an example of your cell. You can actually see the endoplasmic reticulum is kind of wrapped around the nucleus. You can see the dots on it, and that's the rough aspect of the rough ER, which are your ribosomes. Does anybody know what a ribosome makes? Proteins, awesome. You see the Golgi there. There's a couple of mitochondria. Actually, I think there's one hiding on the top up there too. Yeah, there's one up here hiding. You can see inside of the mitochondria, and they are also kind of, they have that extra membrane that's kind of folded a lot. Lysosomes are ones we've not mentioned, but are also considered to be, and then peroxisome, which is down at the bottom. So for the most part, we covered them all. Okay. Mitochondria, you always hear it's the powerhouse of the cell. That's a little bit unfortunate. That's all the people know about it. So it's the powerhouse of the cell. It's more refined than just I'm going to make power. We're going to make power for the cells, more than just ATP production. But it's really interesting. It's got double membrane. Okay, it's the only thing that I can think of in an animal cell that has a double membrane. And a huge part of it is to use oxygen, as we have broken down things from in our food down to glucose, take glucose and turn it into ATP. So one of the reasons why you don't have oxygen, so if you suffocate, okay, you run out of oxygen. And I told you that death actually comes from not having ATP. This is the reason why the lack of oxygen leads to the lack of ATP. Without one, you can't have the other, okay? You can't lose oxygen. Oxygen is actually a final electron receptor in the process of cellular respiration. So it's really important. Interesting notes, because I think general knowledge is nice. Mitochondria are thought to be bacteria that eukaryotic cells absorb at some point. So what we call them an endosymbiont. And so they actually have a lot of factors in them that seem to be bacterial-like. Yet they reproduce inside of our cells. In fact, they reproduce their own cells. The cells don't make them, they make themselves. Interestingly enough, they also contain DNA. I think I mentioned this the other day, right? Where I asked you where the DNA was inside your cell. We say nucleus. It's also found here. Now, is it a lot of DNA? No. But the DNA is specialized for the mitochondrion itself to help it reproduce, to help it to make its own enzymes, to make its ATP. And the last thing I'll mention, just because I think from, I want to connect this to what we talked about the other day, the DNA found here can be used to track maternal lineages. Okay? So if you want to know your mom's lineage, this is the DNA that you would focus on, because this DNA only comes from mom. Nuclear DNA gets mixed up between mom and dad. And I mean, we're not going to get into this in depth, but I'll tell you, mom and dad's DNA can mix like crazy. Grandma and grandpa's DNA mixed coming into your parent, both sides. So it's hard to track one lineage from that DNA. The mitochondria only comes from mother. Okay, we have ribosomes. They are made of proteins and ribosomal RNA, and they are used in protein synthesis. Y'all had this already. They are found in two locations. One is part of the rough endoplasmic reticulum. Why do we even call it rough? It's because under the microscope, when people first identified the ER there, it looked rough. That's the only reason it's called that. It's because the ribosomes are there. So visually, it was rough. Okay. There are also some free floating in the cytoplasm, which probably is not surprising because you need to have proteins everywhere. So your endoplasmic reticulum, perfect lead in. It's a fluid-filled set of tunnels or canals that carry substances within the cell. And they are continuous with the nuclear membrane. Okay, so they're not separate from your nucleus. There are two types, the rough and the smooth. Rough is because you've got ribosomes, as we just said. So the rough endoplasmic reticulum has ribosomes. They synthesize proteins. and then they will transport those proteins within the cell. That's kind of the job of endoplasmic reticulum. And so there are cells that kind of specialize in making lots of proteins, and so you'll see this in high abundance. So you can kind of see that happening over there. So you've got a ribosome over to the far left that's red. You've got mRNA that's being read. It makes a protein. The protein gets folded, and then the protein packages inside of this rough ER into what's called a vesicle, little packaged protein, and then it gets transported. Okay, so this is transport within the cell. Okay, within the cell, because when you get down to step 4, a lot of times these travel to the Golgi and then will get processed further, a lot of times to go outside of the cell, okay, to go from one cell to another or another part of the body. So your smooth ER don't have ribosomes. They are more used to doing lipid metabolism. So for example, breakdown of lipids, because lipids are weird. Okay, why are lipids weird compared to the other macromolecules? Let's think through that again. What's the general words that we use for things that are lipids? Fats or oils, and those things love to be in water, right? No, by no means. Okay, that's my sarcasm. I was trying to see if I could catch it. So can you just process lipids easily inside of a cell? Why not? Why would lipids have an issue? And why would we want to do it specialized, maybe packaged in a certain way? The cells are full of water, at least 60% water. And so you're dealing with a hydrophobic lipid and a bunch of hydrophilic water, hydrophilic water. So you've got kind of a problem working on lipids within a water medium. So the endoplasmic reticulum, the smooth version, allows you to do that in a localized, specialized place. It's also involved in detoxification of drugs and pesticides, so it's pretty important. Okay, the Golgi looks like a bunch of flattened stacks, and they have a bunch of those tiny vesicles coming in associated with them. They basically are modifying and then packaging whatever comes from the rough ER. So you can produce what's called secretory vesicles. Secretory, what do you think is going to happen? It's going to be secreted a lot of times outside of the cell. It's going to also involve in-house proteins and lipids, and then lysosomes. Lysosomes are interesting because lysosomes, when you see the word lyse, L-Y-S, okay, or L-Y-S-E is the word lyse itself. To lyse, something is to basically pop open a cell or some kind of bubble. So we have to sometimes do that. And so you can see how kind of flat and stacked it looks like here. So this pathway 1 here is something that's packaging for release. So it creates a little vesicle, just like we saw back here, which disassociates from the Golgi and then associates itself, kind of merges with the outer membrane, and then allows it to actually secrete things. Here is where we get proteins that need to go on the cell membrane, and they get merged into the cell membrane through this path. And then path 3 is the lysosome. And these are all set up to try to they're going to bind with and ingest other substances, sometimes foreign cells that are invaded, or to kind of destroy, take apart things that we don't need anymore that the cell has made. Slightly like a recycling mechanism. So we've got these lysosomes with bags of digestive enzymes, and they can digest your worn out or non-usable cell structures. They can also act as phagocytes that dispose of bacteria and cell debris. So if you've got living bacteria or dead bacteria, they can take it apart. And if you've got any kind of cell debris of cells, maybe even your own cells that have died, they can break it down. I love the term phagocyte. For some reason, I just think I don't know why, it's fun for me, but phagocyte. There's two parts to this word. Does anybody know what either phago or cyte might mean? So if I said cytology, that would be the study of what, maybe? I'm trying to see if y'all caught it before. Cyte means cell. It just means cell. Phago, if y'all know phago, I'll be shocked. Does anybody happen to have an idea of what phago kind of means? So it kind of means to eat, which maybe is a little simplified version of what that actually means, but this is how I think about it. Phagocytes eat cells. So the process of phagocytosis is all about taking something and taking it apart. So you might think of it as like eating, taking it apart and actually reusing the stuff at other points. So this just shows you my nerdy side. I think I've told you I love Star Trek. I don't know, I think I have. I usually tell all my classes I love Star Trek. And in Star Trek Voyager, there is a species of aliens that have a disease called the phage. See any connections there? And so, in fact, we even have viruses that attack something. Can you imagine what those viruses attack and hurts? They're bacteriophage. They attack what kind? Bacteria, yeah. So these are like bacteria eaters, even though they don't actually eat, but they attack it. So in that Voyager episode, they had this disease, and it was called the phage. And the phage would actually slowly like take apart their bodies. And so they would go around the galaxy, well, their quadrant technically, and they would steal body parts from other aliens and graft them onto them to replace, which never got rid of the phage, but only just prolonged their ability to live. Maybe a little bit gruesome, but that was one of the things that happened during the Voyager season. So that's one of the reasons I was able to always remember this, because I had that picture of, these like pieced together aliens, because different parts of them were getting eaten off by this disease. So bacteriophage, phagocytes. So you have something within your cells that can destroy invading bacteria or take apart cell debris. I don't know about y'all, but I find that pretty exciting, because otherwise, how do you not fill up with trash? With broken down cell parts that get messed up, or you made too much of something. How do you take away those things? We have a recycling system, and phagocyte is one of the ways to do that. So I mean, every time you get an infection, what happens to the actual cells that infected you, or the virus that infected you? Just hangs out there for the rest of your life? No, you take it apart. Is that exciting for y'all? I don't know. You can tell I'm a nerd. I think it's so much fun. I think it's kind of an awesome system that's been put together. Okay, peroxisomes. This is maybe the one that's talked about the least. They have oxidase enzymes. And I'm just going to stop right there and make a connection to the name. Do you know of any chemical that we might keep in our bathrooms that looks similar to peroxisome? Hydrogen peroxide, H2O2. Okay, and those things can oxidize, right? Um, Little side note, because I'm always full of information that you may or may not need. What happens when you put peroxide on a cut? It bubbles. Should you be putting peroxide on a cut? The answer is no. Why? Because peroxide oxidizes things, not only the infection, but your cells. It can actually slow down the healing process because you have not only destroyed whatever might have infected you, but also the cells that are at the edge of the cut as well. So why is it bubbling? Because that's the reaction. Does anybody know what the bubbles actually are, like gas that is? What can come out of that? You could get hydrogen. That's not the gas. That could be fun, flammable. Not really. What else could you pull this out of? It's not going to be straight hydrogen. It's a good guess because you only have like 3 choices. That was 1 of 3. The other 2 are right. What else? What else could this make? Yep, the gas is actually oxygen. Both of which have no issue for you. Okay. So hydrogen peroxide needs to be broken down. Okay. I should clarify a little bit that it's not the process of H2O2 does not actually make this. The H2O2 ends up just oxidizing with other stuff. What causes this is an enzyme that your body has to break that down so it doesn't damage it. Okay. So that enzyme, when it breaks it down, is trying to get rid of this as fast as possible so you don't get hurt. Because these things don't hurt you. That's why you bubble. Not everything has that enzyme. There are some bacteria that don't. And in microbiology, we will test if certain bacteria bubble or not with hydrogen peroxide. If they bubble, then we know they have an enzyme to break it down. And of course, they have some protection against it. For those that don't bubble, don't have a protection against it. So, interesting side note. Why do I bring that all up? Because we're dealing with something similar here in the peroxisome. So it detoxifies harmful substances like alcohol and formaldehyde. It breaks down free radicals, which you do not want in your cells because they're highly reactive, sometimes can be cancer causing. Free radicals, they're converted to hydrogen peroxide. So there is a connection here, which then has to be converted to this. This is a byproduct to some degree, or an intermediary product, if you want to think of it that way, before we get to something that's less harmful. Okay. And they get made from the ER, which is not all surprising if you think about it, because most of these things have some kind of pinching off from the ER or the Golgi. Okay, cytoskeleton. Okay, what do I want you to know from this one? We are not going to go into great detail with this, but what I do want you to know about the cytoskeleton is that you have a network of these are proteins, and they're all throughout the cytoplasm, and they create an internal framework that gives you the shape of your cell. It supports the organelles, provides a way to transport things throughout the cell as well. Okay, I'm not going to ask you to know microfilament versus intermediate filament versus microtubules. Okay, so don't worry about that. I just want you to know the cytoskeleton. Here's what they look like. So actin subunits, we'll probably bring up that again at some point because those are used in muscle movements. So just know that the cytoskeleton will be involved there. our ability to move things within ourselves, but also to move our cells themselves, require us to know how these things work. But we don't need to go into great depth. Just know that you actually have a framework inside of your cell that gives some structure. I like to think of it as in some ways it reminds me of, those roller coasters that you hang off of. So like you sit in and they have the contraption to hang up here and then you go zipping around. That's kind of how I think of the transport sense, some of this. Does that make sense from a visual perspective? Do you know what I'm talking about? I don't know. I can only think of one roller coaster that I don't think y'all know. I don't think y'all know this roller coaster. Do y'all know what I'm talking about at all? You know what I'm talking about? Yeah, so it hangs off of. Yeah, so when you see it, you kind of see it kind of get thrown like this around. Yeah, I think the Batman is an example. Yeah, I'm not a roller coaster person. The last time I was on one of those, I was in high school. So I don't even think the one I was riding, I'm not even sure if it still exists or if it has the same name. This is not something else, okay, that I want you to worry about are the centrioles. I just want to show you that where's some places where these are connected, but I'm not going to test you on these things, okay? So centrioles are microtubules, examples of your cytoskeleton. They're used in mitosis. So at least in one place. We haven't talked about mitosis, but do y'all know what it is? Cell replication. Okay, it's the way that you make more of your cells, and they're copies of those cells. So it gets used there during that division process. It helps to control where that DNA goes. Where does it form first, and then where does it get moved to? Okay, so there are different parts of your cytoskeleton that attaches to the cell membrane. Sorry. Sometimes it can change shape. Now, so this diagram here is just a little recap of some of the stuff we've covered. So the plasma membrane, there is the cytoplasm, organelles, the ribosomes, rough ER, smooth ER, Golgi, peroxisomes, lysosomes. So when you're studying those things, kind of puts it all in, not all the details, but it kind of gives you some framework to kind of visualize and see this in a little table format. And then you don't have to worry about this one, okay? Here are, so the nucleus, nuclear envelope, nucleolus, chromatin. You do need this. Okay. Check something real quick. Still not quite where I want to be at today. Okay. You do want to know about surface extensions, things that are off of the cell, because these are really important. So you have one thing called cilia, and these move materials across your cell surface. If you wonder why your mucus can travel within your body, it's because of the cilia. The mucus inside of your respiratory system gets moved with these little hair-like extensions. They're very hairy-like. Flagella propel cells from one place to another. Many bacteria have flagella. In humans, only the sperm has a flagellum. Okay. So flagella is plural, flagellum is singular. Then you've got microvilli, and basically this is, I would say, your cell membrane has been folded very nicely. Okay, so it's just extensions of that plasma membrane. What this does is it gives you more surface on that cell. This one's important because places like your small intestine have great amounts of microvilli. The purpose is to increase the surface area so you can absorb more nutrients after you break down all your food. So there's our flagellum. There really is not much going on inside a sperm cell. There's not a lot of space. Look, the nucleus takes up the majority. Because what's the purpose of a sperm cell? Just take DNA. That's it. Okay. There's not even mitochondria in there usually. Because these are not supposed to last long. So could you get a mitochondrion from your father? Possibly. Could a sperm have one in it? Yes, it's a possibility. Is it likely? Less likely, much less likely. Okay. Critical thinking question. If the sperm cell that joins with the egg to form a person, if that sperm cell has a mitochondrion in it, maybe one, two, whatever, if there's enough space, what would that mean for that person in terms of their mitochondrial DNA? So would that person have mitochondria from their father?
But it's one like one mitochondria is gonna overpower
About what?
because the mother would have more.
Absolutely. Egg cell, actually the egg cell is the largest cell in the human body. Okay. And it is big enough to see, just from the dot of your pencil, like it's large enough to see with the naked eye. So even if it brings a mitochondria or two, compared to the amount of mitochondria that the mother's given to that egg cell, which is way more than normal because egg cells don't evenly divide when they're produced. So one cell gets much more than the others. The mother's mitochondria are going to be more important in terms of how much DNA is in that cell compared to the father's. So even if it did come, there would be DNA from both. But just on the situation of dealing with numbers, the mother's going to have more. Now, it's true. You would have DNA, the mitochondria from both parents there. Let me ask you this. Would all cells in that new person have mitochondria from both? Ah, okay. Think about this. So you've got a large egg, and you've got this sperm. Okay. They combine. What do they combine? We're totally getting ahead of ourselves, but these two are going to form one cell, right? With a full complement of DNA. Okay, it's called a zygote. It's one cell, and you were created from that one cell. So if mitochondria went in, mitochondria was already here, would this zygote, if every cell formed from it, have the same mitochondria? Yeah, but the amounts may not be, like the proportions between the two may not be the same.
For every single
Cell in the,
cell, I mean, every single one would be. I would think the mitochondria, you would only one mitochondria. You would have enough to go around.
so that's a good question. You made a point that I didn't think about. So let's say it has one, comes over here, it replicates a few times. Is there a possibility that some of the mitochondria, like the mitochondria that came from here, didn't make it? So if this is one cell and then it goes through and divides, would it divide evenly where some of those, could it possibly just have made it to this side. Yeah, that's a possibility because the number could be so small. Now, we have the cytoskeleton where things can move around. Is it possible that some of these that were formed, even if it's just a few, got on this side? Yeah, so it could end up in all cells and maybe not, depending on how few there are. So yeah, that's a good way to think about it. And that's kind of fun critical thinking to think through. As you learn new things, like, how would that look? So if someone did a DNA test for someone who had this happen, they should actually have two sets of DNA to be found, which could be confusing for the people who are running those samples. But we can do it because it's very unlikely to happen. That sperm usually doesn't carry it, at least in humans. We won't get outside of humans because you want to have some fun in biology, go find something that does everything against all the rules you ever learned. And they do exist. Plants being the weird ones, especially. Okay, that was just kind of a fun thing. So you've got a bunch of different cell types. We're talking about these cells that we're making and what's inside of the cell. You've got over 200 different types. They're pretty small, 1/12,000th of an inch, at least on the low end. And then you've got some really long nerve cells of a very different shape. So shapes actually give you different functions. They're held together in different ways. We talked about how they can be held together. So part of being a multicellular organism is that while all of our cells have the same DNA, not all the same DNA is turned on in them. And so they get specialized for different functions. In your heart cells and in your brain cells, you have the same DNA, right? But they don't do the same thing because different genes have been turned on inside. Okay. So what can cells do? I think this is pretty self-explanatory. They can metabolize, they can digest food, they can dispose of waste, they can reproduce, they can grow, they can move, they can respond to stimulus. Basically, anything that an organism can do, they can do, right? And I think I'm going to stop there because I think that's a nice stopping spot. Do y'all have any questions for me? I haven't got your quizzes graded because yesterday did not go like I was hoping. I ended up being way busier than I really thought was going to happen. By Friday, for sure, I might have them graded. I am really close to finishing this PowerPoint. I don't know if we'll finish it Friday. So I want to talk to you really briefly. I'd like to have test one next week. But would there be a specific day that's better for you? I don't think you'll have something else planned test-wise.
It's not on Monday.
Just not Monday?
Not Monday.
Not Monday, not Monday, not Monday. Okay.
You do any other day, not Monday.
I mean, we can shoot for, what do y'all think, Wednesday or Friday, in preference? I will tell you this, Friday doesn't give you more material, because when I finish this PowerPoint, we're done with material for test one. So if we finish it, which I think we'll finish, if we don't finish it Friday, we'll definitely finish it Monday. So if we did it Friday for the test, you could have a little more time to study. And if I need to, I'll just start in on new material, but you can kind of check out a little bit. That's what I'm thinking. Do y'all like doing the quiz in person on paper?
Yeah.
Well, when I give tests, especially for things like this, I like to have both a written portion and a multiple choice portion. So test format is going to be a majority, maybe like 2/3 multiple choice, and then we'll have some written portions. I think if we do it Friday too, that will also give us a little bit of time to review and prepare. And so we might take either part of Monday when I finish, or sometime on Wednesday to be able to do that. I am a little bit later than I planned in the syllabus in half test one, so we're a little bit past that. But let's do it next Friday. I'll have your quiz done for this Friday so we can talk about it. We'll probably be finishing this on Monday. We can spend the rest of Monday reviewing kind of what to do. Because there's a lot of material, but some of it is not going to be testable. And I want to show you some things, but not so testable on some of those things. Curiosity? Okay. Have a good day. I'm like 20 seconds from being on time. Unlike my normal over about 3 or 4 minutes. I'm so sorry, guys, that I do that.